genomic future inbreeding, GFI, and maybe had a question about what that really meant. I'll just ask Kurt to kind of explain that.
Well, it's, I'm not certain because I don't calculate them, but I think it's just using the type of data that we're looking at in the figure that I showed, which is essentially the fraction of alleles in common across pedigrees and using that rather than the pedigree-based information. I fully expect that within a year or two that will become de facto standard and EFI will cease to exist. If I can add a couple other comments. To Jeff's first one that will keep track of lines, we currently do, they're called breeds. And so the easiest way to control inbreeding is to crossbreed. And that is somewhat tongue-in-cheek, but it's also the other tool that Genomics will give us is the ability to do, and this is something that's fascinated me for a long time, is create a new synthetic breed that's a function of whatever breed people might be interested in. And I think that will become much easier as genomic tools become available. And we're able to find the parts of the Jersey cow that are optimal over parts of the Holstein and so on. The other thing is, one of the challenges is trying to identify what a true outlier or an outcross is. And I think DNA is going to help us identify that. There's currently some tools that we're investigating internally that should help us to identify what, in graphical and terms that everybody can intuitively understand, what makes an animal more closely related to the breed as an average and what makes things a little different. And one of the problems we have is the ability to predict those animals genomically is related to how far from that genetic center of that population they are. And so not only is it important from a diversity management perspective, it's also important from genetic prediction that we have those animals represented in a group of animals that we genotype and have performance data. So I think we need to not only identify the tools that allow us to manage diversity, but also encourage people like Jeff and Julian to progeny test those animals that we identify so that we can better predict and identify the genomic merit of those animals.
Yeah, the point kind of being on this is that in the past we always assumed animals with identical pedigrees, in other words, full siblings, were related to the rest of the breed identically, and that's not true. Some are inherited different genes that cause them to be more or less related than their full siblings.
And actually, the one final comment I'll make in response to Jason is, don't lose any sleep over genetic defects, because we can nail them with this technology with 6 or 7 animals and non-affected sibmates in a heartbeat. So genetic defects will quickly become things that we can eliminate with almost no cost.